Hello everyone. I would like to thank you for joining us today for this webinar hosted by the International Society for Experimental Hematology. My name is David Yudovich. I'm a PhD student with Dr. Jonas Larsson at Lund University, where we, much like the majority of today's webinar audience, are interested in applying the CRISPR system to the study of hematopoiesis, the hematopoietic stem cell, and genetic regulators thereof. Previously, I received the Bachelor's of Science and Engineering degree from Duke University and later worked as a research assistant with Dr. Benjamin Ebert at Brigham and Women's Hospital in Boston. There I was fortunate to take part in work led by Dr. Dirk Heckel on generation of mouse models of myeloid malignancy with combinatorial genetic lesions using CRISPR-Cas9 genome editing. I will be moderating for today's session titled How to Get Your Bloody CRISPR-Cas9 to Work, Tips and Tricks from Experts in the Hematology Field. We have a great three speakers for you today, Dr. Daniel Bauer, Dr. Daniel Bauer, and Dr. Michael Gundry, all with tremendous experience in adapting and translating the transformative CRISPR system to the study of hematopoiesis. But first, a quick note about questions. If you have a question at any time during the presentation, please type your question in the question box or chat box on the screen. We will have time for questions at the end of the webinar, but you are welcome to enter your questions into the chat box at any time during the presentation. With this, it is my privilege to introduce our first presenter for today's session, Daniel Bauer. Dr. Bauer is a physician scientist whose research integrates genetic, epigenetic, and functional genomic methodologies to understand the determinants of blood cell development and develop innovative therapeutic strategies for blood disorders. By comparing common trait-associated genetic variation with epigenetic modification of primary human erythroid precursors and applying genome editing technology, he identified an erythroid enhancer element of the BCL11A gene that is a critical determinant of fetal hemoglobin level and a potential therapeutic target for the beta hemoglobin disorders. His laboratory has developed techniques to perform genome editing using CRISPR-Cas9 RNA-guided endonucleases in hematopoietic cells. He has developed a method termed Cas9-mediated in situ saturating mutagenesis to determine at high throughput and high resolution the function of non-coding elements in their native chromosomal setting. His clinical work in pediatric hematology focuses on the care of patients with hemoglobin disorders. He received his Bachelor of Science in Biology from Brown University and MD PhD from the University of Pennsylvania. He completed clinical training in pediatrics and pediatric hematology oncology at Boston Children's Hospital and Dana-Farber Cancer Institute. He is a principal investigator and staff physician at Dana-Farber Boston Children's Cancer and Blood Disorders Center, assistant professor of pediatrics at Harvard Medical School, principal faculty at the Harvard Stem Cell Institute, and associate member at the Broad Institute. I will now turn the presentation over to Daniel.
Great. Thank you so much. So I'm going to give you a brief introduction to some applications for CRISPR for hemoglobin disorders and hematology. So in this post-human genome era, we have the fortune of having many GWAS studies which have identified genetic variants associated with a wide range of human phenotypes and disease predisposition. But most of these genetic variants actually do not affect protein coding sequences, but rather are distributed in the non-coding genome. And more recently, it's been appreciated that these GWAS SNPs tend to be enriched in putative regulatory DNA. For example, on the bottom left in DNase1 hypersensitive sites, and the bottom right in enhancers. And so this has led to the hypothesis that much genetic variation acts by influencing gene regulation and cis elements. But this is a slide I took from a recent review by Bill Kalem, where he was critically evaluating the cancer target discovery, but I think many of the same issues are relevant as one tries to assign function to the genome. That much of the data, for example, regarding chromatin marks at regions of the non-coding genome are essentially correlative with genetic variants linked with disease. And then if one wants to move to questions of necessity or sufficiency, one needs to use different experimental methods, and genome editing can be incredibly useful here to ask the necessity for sequences for a given function or the sufficiency of those sequences for a function. And so there's really many modes of genome editing one can contemplate, and it needs to be tailored to the experimental question. And so just to discuss a few of these, one can use just a single guide RNA to target the Cas9 nuclease to a given locus and produce cleavage, which then gets resolved by the non-homologous end joining machinery to achieve small indels. And so one can imagine targeting a given chromatin mark or a transcription factor binding site. Another mode would be to use multiple guides to generate a pair of cleavages, and in this case one could have the goal of achieving targeted deletion of a segment or alternatively inversion of a segment or even translocations if the two cuts were on different chromosomes. Another approach that I'll come to in a few more slides is a tiling guide RNA approach or saturating mutagenesis, where one generates a large number of mutations to identify mutations associated with a phenotype of interest. There are a number of approaches that use nuclease inactive versions of Cas9, so-called dead Cas9 or dCas9, that are coupled to various biochemical moieties that may result, rather than in genome editing, in what's referred to as epigenome editing. And so one can imagine using a repressive domain like the Crab domain, or a repressive chromatin regulator like LSD1, to target to a specific sequence using dCas9 and execute repressive chromatin marks. In contrast, one could use a transcriptional activation approach using a variety of activators depicted here are VP64 and P300. And unlike the previous approaches, which are disrupting a sequence and asking about its necessity, these are approaches that can be associated with the gain of a chromatin mark and ask for the sufficiency of that chromatin state for some gene regulation or biological phenotype. Finally, there's a variety of repair modes that may involve homology-based repair, non-homology-based integration of an extrachromosomal cassette, or even base editing maneuvers in which sequence bases may be changed in the absence of cleavage per se. I wanted to make one point about paired double-strand breaks, because this is often a definitive mechanism to delete a given element. And so what happens here is, as shown with these two sgRNAs A and B, that there's a pair of cleavages, and then there's actually 4 ends of the DNA that the non-homologous end joining machinery can act on to paste back together. And so although the intended consequence may be to have deletion of the intervening segment, alternative outcomes are also likely, including multifocal indels at each of the 2 cleavage sites, or inversion where the intervening segment is flipped. And so when we investigated this in hematopoietic cells by introducing a pair of guides at a number of loci, we did find we could achieve deletions at a reasonable rate, about 25% of the alleles. But since these sort independently in cells, what we found was only a small fraction here, 11% of the cells had biallelic deletions, and in a population of cells, most of the cells had complex outcomes, including inversions and what we would call scarring or multifocal indels. And so this can limit the efficiency of achieving the desired target and needs to be taken into account when designing a genome editing experiment. So I want to give a brief vignette based on our interest in fetal hemoglobin regulation. This is because fetal hemoglobin is an alternative form of hemoglobin whose high expression can ameliorate disorders of adult hemoglobin like sickle cell disease. And in this case, the GWAS was really quite informative in that looking for common genetic variants associated with fetal hemoglobin level just identified 3 loci that explained a huge proportion of the heritable variation in fetal hemoglobin level. And highlighted here in this yellow shaded box are variants on chromosome 2 marking a gene B7A. And so if we look at where these variants are with respect to the B7A gene, like many GWAS signals, these variants were not affecting the coding sequences of B7A, and they clustered in this distal second intron. But what was striking was that erythroid precursors had peaks of DNase-1 hypersensitivity overlying these trait-associated variants. And so zooming in, we could see there's 3 discrete DNase-1 hypersensitive sites listed here as 55, 58, and 62, which is based on their distance in kilobases from the transcription start site, each of which had overlying trait-associated variants. And other tissues which express high levels of B7A, like brain and B lymphocytes, had a paucity of this chromatin mark suggesting that this could be erythroid, these sequences could have erythroid-specific regulatory potential. So this led us to a hypothesis that perhaps if one could collect blood stem cells from patients with hemoglobin disorders and disturb this B7A erythroid enhancer, one might achieve disruption of B7A, elevation of fetal hemoglobin, and potentially ameliorate the patient's condition. And in fact, when we use this paired double strand break approach to delete the enhancer from erythroid cells, we found, yes, indeed, in the absence of the enhancer, cells lost expression of B7A and had very high expression of fetal gamma globin. But the question we wanted to ask was, would single genomic cleavages, indels, just disrupting minimal critical sequences within this enhancer result in robust fetal hemoglobin induction? And so to ask this question, we took the approach of conducting an agnostic screen. And so I want to go through the design and the performance of this screen. So this is drawn to linear scale for a prototypical 100 base pair enhancer element, and the design of guide RNAs is restricted by the presence of the protospacer adjacent motif, which in the case of Cas9 is NGG. So in a 100 base pair stretch, there's about 13 of these PAM motifs. So then we could design 13 guides on average per 100 base pair stretch. And so what would happen would be in a population of cells, any one cell getting one guide would have cleavage and then would variably repair. So some alleles would have 1 base pair, 2 base pair, 3 base pair, so forth, generally 1 to 10 base pair indels around this cleavage site. So in response to a single guide RNA, in a population of cells, there's an allelic series of indels. And then by tiling many guides throughout the target sequence, one gets a series of allelic series that, given the expected indel pattern of 1 to 10 base pair deletions, can approach saturating mutagenesis in situ, disturbing almost every position within the enhancer or target sequence. So the way we did the experiment was to order the oligos as a pool, clone them as a pool to a lentiviral library, then transduce cells at low multiplicity. And this is key so that any one cell only receives one guide RNA integrated into its genome. And then we're able to culture the cells, stain them for fetal hemoglobin level, and then by flow cytometry, physically sort out cells with high or low fetal hemoglobin level, isolate the genomic DNA from those cells, amplify the guide RNAs from the lentiviral integrants, perform deep sequencing to measure the representation of individual guide RNAs, and then map those guides back to the genome to produce a functional enhancer map. And so this is the kind of results we observe from the screen. For each guide RNA, we calculate a fetal hemoglobin enrichment score, which is merely the number of reads in the high versus low fetal hemoglobin pool. And you can see, starting on the left, a set of negative control non-targeting guides showed no enrichment, and a set of positive control exon targeting guides intended to create null frameshift alleles of B7A demonstrated positive enrichment, showing the capability of this screen to identify guides that disrupt B7A and lead to high fetal hemoglobin. And then looking at these three DNase hypersensitive sites, 55, 58, and 62, one can see that most of the guides showed no enrichment, which is consistent with the hypothesis that usually producing small indels and non-coding sequences would be without functional consequence. But what we observed was stripes of co-localizing guides of shared functional activity at each of the hypersensitive sites, with a particularly robust stripe at this plus 58 site, where we found 10 guides who specified cleavage within 42 base pairs whose enrichment exceeded that of the median exon targeting positive control guide. So basically, in short, this screen was able to ask for many guides, if any of them were functional, and identify a set of guides who created mutations in a narrow region of the enhancer, indicating critical functional sequences within the enhancer. And so this is another way to show that data, zooming in and listing two of these top scoring guides here. And so again, the way that we calculate these scores is based on the enrichment of the guides themselves. But another question one would ask is, what are the mutations that these guides make, and what are the mutations themselves that are associated with your phenotype, in this case fetal hemoglobin? So to do that, to ask that question, what we did was turn to an amplicon deep sequencing approach, where we amplify our target sequence, and we compared cells isolated from a high and a low fetal hemoglobin pool, and then perform deep sequencing of the amplicon to ask what are the indels specifically associated with elevated fetal hemoglobin. And to do this, we received help from our computational biology colleagues Luca Pinello and GC Yuan, who developed this tool, CRISPR-based, that can take in deep sequencing data and quantify and visualize a variety of genome editing outcomes. And what we found here is, by plotting the data on a per nucleotide fetal hemoglobin enrichment score, that the indels that were most highly associated with fetal hemoglobin mapped to this positions intervening these top two scoring guides, and underlying which was this motif for the erythroid transcription factors GATA1 and TAL1. All right, so just another note here is, these are the guides. You can see between these two guides there's about 50 base pair gap, and that's just due to the absence of NGG PAMs at this sequence. So the resolution of this sort of an assay depends on the distribution of PAMs in the genome. So how can one improve on that? Well, there's a variety of Cas9s, including those that are engineered to have altered PAM specificity. There's different Cas9s from different species of bacteria that have different naturally occurring PAMs. And then there's even non-Cas9 CRISPR nucleases like CPF1. So here's an example of a gap where there's about 115 base pairs between cleavages as specified by NGG restricted forms of Cas9, but by using an NGA engineered variant Cas9, one can fill in the gaps, and reciprocally a gap due to a paucity of NGA sequences can be filled in using an NGG nuclease. we have extended this saturating mutagenesis approach to look at other loci implicated by GWAS, including that at HBS1L MIB, where there are variants associated both with fetal hemoglobin level and other erythroid traits. And although this is a very heavily chromatin-marked region with DNA sensitivity, transcription factor binding, and even super enhancer presence, what one can see is the non-coding genome around this GWAS signal is littered with repetitive elements. And so this is a major challenge that I want to highlight when using CRISPR to target the non-coding genome. And so it turned out when we just blindly designed all guides around this GWAS marked elements, we found that many of the guides had multiple genomic matches, up to hundreds of thousands even of genomic matches due to the frequency of repetitive elements of these sequences. And that many of these guides caused cellular toxicity in a fashion that was not dependent on the actual requirement for sequences at MIB, but likely just due to frequent cleavage of the genome. So I think this highlights that when one is targeting the non-coding genome in particular, stringent filtering of the guide RNAs is needed to avoid potential off-target effects. Finally, there's naturally occurring genetic variants. In fact, that's the basis of GWAS. And so generally one uses the reference genome to design guides, but your cell type of interest, your donor may have SNPs that either decrease the efficiency of guides cleaving those target sites, or may even block the recognition of Cas9 by destroying the PAM, and may even create new PAMs that allow for the design of new guides. So we and others have made algorithms that can help one design guides that are restricted by a variety of PAMs and that can take into account naturally occurring genetic variations. So in conclusion, there's a variety of modes of CRISPR that I think are really useful, including individual guide mutagenesis, targeted deletions, tiling guide RNAs, and in something I didn't have time to talk about, CRISPR inhibition, which can help determine the necessity of genomic sequences in their natural chromatin context for a variety of biological functions. And that one needs to carefully select both the guide RNAs and the nucleases to maximize the genome targeting space to minimize the off-target effects and account for genetic variants. So I'd like to thank my collaborators, colleagues, funders, and your attention, and I'm happy to take questions at the end of this session.
Thank you so much for the wonderful presentation, Dr. Bauer. We now move to our second speaker, and I have the privilege of introducing Daniel Bauer. Dr. Daniel Bauer is a research instructor in the laboratory of Dr. Matthew Porteus at Stanford University in the Department of Pediatrics, Division of Stem Cell Transplantation and Regenerative Medicine. He completed his PhD in molecular toxicology at the University of Rochester, where he studied the mechanism of the aryl hydrocarbon receptor in mediating cerebellar transcriptional programs. Daniel continued his academic track as a postdoctoral scholar in the laboratory of Dr. Yoon J. Cho at Stanford, where he studied the molecular mechanisms underlying cerebellar tumor formation and progression, with a particular focus on epigenetic dysregulation. Because he was interested in genome editing, stem cell biology, and hematology, he decided to make a paradigm switch to Dr. Porteus's group, who specializes in hematopoietic stem cell gene therapy. During his postdoctoral work in the Porteus groups, they developed a CRISPR-Cas9-based beta-globin gene editing by homologous recombination methodology in CD34-positive hematopoietic stem and progenitor cells as a potential therapeutic strategy to treat severe sickle cell disease. Details of these studies are described in their article in Nature, and Dr. Bauer is now leading IND-enabling preclinical efficacy, feasibility, safety, and tumorigenicity studies for FDA approval of a first-in-human clinical trial at Stanford for the treatment of severe sickle cell disease using CRISPR-Cas9-based beta-globin gene correction. I now will turn the presentation over to Daniel.
All right. Okay, great job, Dan. That was a good introduction to his lab and what they're doing at Harvard. And so what I thought I'd focus on today is the should and the should nots of CRISPR-Cas9 gene targeting. So most of this is going to be about genome editing by homologous recombination strategies. And so what kind of the dogma within the Porteus lab is co-opting the endogenous homologous recombination pathway to correct gene function in hematopoietic stem cells for the treatment of serious diseases. So shown here is the kind of paradigm that Dan's kind of already gone over is, could we be able to isolate stem cells from patients and deliver a nuclease, so in this case Cas9, and also a homologous donor template, in our case by recombinant AAV6, and be able to change bad genes for good genes and then infuse these corrected stem cells back into the patient. And so we are lucky enough to be able to do this particularly well at the beta globin locus in hematopoietic stem cells last year using this gene targeting approach. And the details in that, again, can be found here. But I'm going to be focusing really on kind of the nitty-gritty of the approach as the audience here is, researchers looking to do homologous recombination. So I thought I'd go through those steps in order. And so I kind of broke down the webinar into three main points. One is generating gene targeting reagents to be able to do homologous recombination. The second is actually performing the gene targeting experiments. And the third is analyzing these editing outcomes in vitro or in vivo. And so first we'll focus on generating the gene targeting reagents. So the main things you need for editing of hematopoietic stem cells by homologous recombination, of course, one is human CD34 positive HSPC, so I'll go over the different types of sources and their efficiencies and also their potencies. And two is, within our lab, we use chemically modified synthetic guide RNAs. We found that they're highly active within stem cells in particular. And again, I'll go over those type of modifications. And three is using a recombinant SP Cas9 compared to other sources of Cas9. And I'll go in a lot of detail about how to design a homologous donor template and package it and be able to make AAV6 to deliver your donor template. And lastly is, of course, you need a way to deliver the nuclease. And in our case, we use an electroporator. So I'll go over the type of electroporators we use. So human CD34 positive sources, there's a variety of sources. I made this table, and over here on the left is showing the different types of sources. So I broke it down in the source type, the engraftment potency, the targeting efficiencies in our hands, and what I've gathered from the literature, and of course cost. And so cord blood has the best engraftment potency. In our hands, it leads to high targeting and efficiencies, but the unfortunate thing is it costs a lot from commercial vendors. I put an asterisk here because a lot of places have cord blood programs that combine, the cord bloods from birth to the research facilities, and so that obviously makes it much cheaper and is highly recommended. And then there's bone marrow, which has great engraftment potency, but in our hand has less targeting efficiencies for unknown reasons. Mainly, my guess is because of the cycling, and the costs are less than cord bloods. And then there's mobilized peripheral blood and non-mobilized peripheral blood, which have a better or worse potency than the other two I described, and the cost gets cheaper. And so I think the big thing here is you just choose your 34 source type with your research interests. So once you have 34s in hand, then you need a guide RNA. And I know Michael's going to be talking a lot about guide RNAs and knockouts, and so I'm not going to go into that in too detail. But in our lab, we use a chemically modified guide RNA, and the one we use is this MS. So it's modified with 2-O-methyl and the thiosulfate or the phosphorothioate to prevent immune stimulation and also to prevent exonuclease degradation within stem cells. And we had a paper in Nature Biotech showing that these were very active in primary stem cells. And so we order these guide RNAs from right now either Synthego or TriLink, and of course this study was in collaboration with Agilent, but if you're trying to do for research scale, small scale is within Synthego and a medium scale is within TriLink Biotechnologies. So we recommend buying these synthetic guide RNAs that again have these modifications at the 3 terminus within the 5' and within the 3'. And so once you have a guide RNA, of course, then you need the Cas9 nuclease, and we use the Streptococcus pyogenes, the SP Cas9 within the lab. And again, I made this table about the different ways to generate or to use or deliver the Cas9 nuclease. I know Michael's going to go into a lot of detail about the RMP, but I just wanted to show this table showing you can either deliver it by a recombinant protein via mRNA or lentivirus or plasmid. And they have different efficiencies, and they also have different on to off target ratios and different toxicities. Of course, we use the RMP because it has high efficiencies. It's kind of this hit and run editing approach where you have high and you're on, and you don't have a lot of nucleus exposure to get off target activity. And the toxicity, because of the duration, is very low. And of course, plasmid is the lowest on all these, and I don't think the field is using plasmids anymore anyway. But we recommend using RMP, and I put some vendors down here. We get it from IDT Thermo. I know UC Berkeley selling it, and I just saw Jacob Korn put a protocol online to make it in-house, which of course would be very cost effective. And then next, you need a donor platform to deliver your homologous template. And so within our lab, we use recombinant AAV6. And so data that came out of Paula Cannon's lab and Sharon Berg's lab and Rawlings' lab and Harry Malick's lab showed that this donor was highly efficient within hematopoietic stem cells. And there's a lot of history of AAV being able to mediate homologous recombination in a variety of cell types at high efficiencies even without a double-strand break. And so this table was kind of to show the different donor deliveries, the efficiencies, and what kind of modifications it can make. So SNP here is, can it make the resolution of single base pairs? And gene, can it put it in whole gene cassettes and toxicity? And we think AAV kind of satisfy all those criteria that we're looking for in a homologous donor template. So I thought I'd spend some time going over how to actually make a homologous AAV6 donor template, at least to be able to clone it and design it. And so we use Gibson cloning within the lab, which is, I highly recommend it. It's a great way to stitch pieces of DNA together from different plasmids without using restriction enzymes. And so shown here is the genomic locus, and I'm going to talk about 3 different ones: a reporter donor, a SNP donor, and trying to knock in a cDNA donor. So the reporter donor, you have shown here, you have your break site between 17 and 18 base pairs and the guide, and 400 base pairs of homology flank that site. And so if you want your homology arms as close to the break site as possible, and preferably within 10 base pairs is the kind of rule we follow in the Porteus lab. And 400 base pairs is the smallest we found that mediates efficient HR. You can go higher, but we haven't really found it helps a lot. If you go lower, it can hurt, especially below 200 base pairs. And so a promoter here, and so showing the donor here, so you have your 400 base pairs of homology on either side of the break, then you have a promoter driving a fluorescent protein with a poly A. So promoters, we recommend S of FV for kind of a non-clinical or high-expressing viral promoter, or UBC as your kind of clinical or non-viral promoter that leads to sustained expression. And for fluorescent proteins, we recommend GFP. And if you want a cell surface marker to use for flow cytometry, we recommend the truncated NGFR receptor. And poly A's, we usually use the BGH poly A. And so that would be the recommended way to design a reporter donor. But a lot of people want to be able to mediate SNPs. We use the AAV6 donor to mediate SNPs. We know that you can use the SSODN, but we think within our hands it's a little bit more efficient. But we recommend testing that in-house if you're working with SSODNs right now. And so for this, there's one rule, though. You want to make sure the packaging is over 2.5 kb. Of course, there's no promoters here, and there's no cDNAs, and you're just mediating small base pair changes. And so we make the homology arms 1.2 kb on either side of the break. And what's shown here is the genomic locus and then the protein coding regions shown up here with these amino acids. And what you have to do to make sure is that showing the break here, you want in your target change over here, you want to diverge from your break to your target region, the coding region. And so you do that because sometimes you can get premature crossing over if you don't change the nucleotide sequence between the break and your region that you want to change. So essentially it'll look like a homology arm. And so here is showing these stars what you'd want to change within these coding regions, but to maintain the same amino acid code before you get to your change from X to Y. And so following homologous recombination, you'll have your Y change, but the rest of the amino acid code will stay the same. Another application people want to use this for is to be able to knock in cDNAs into the endogenous locus to drive, or in this sense, to correct a gene function. And so lastly, just showing here, you do have the same base pairs, your 400 base pairs of homology, but the difference here is that your one homology arm is right next to the break site, but the other homology arm is next to the ATG. And shown here in white is the distance between the ATG and your break site, and that could be as much as 100 base pairs. The farther this goes away, the reduced efficiency it gets, but it is possible as long as one arm is close to the break site. And I also wanted to say, you should really determine for your gene of interest if you need early intronic sequences necessary for equivalent mRNA production. Some genes require intron 1s for high transcript production. And so once you have your donor designed using those methods, a big thing is to be able to produce the AAV in-house. And the way we do it is the triple transfection method in HEC293 cells. And I put some down here, some references for some nature protocols out of Jude Smolsky's lab and David Russell's lab that are really good that we used to follow in the beginning. And so the basic scheme is that you have your vector plasmid, which again you would clone in what we just described between ITRs, and then you would have two plasmids, one that delivers your helper plasmid, so the rep and the cap. So in our case, it's going to be rep2 and cap6, and the adenoviral helper plasmid will be ad5. Shown here is the triple transfection method, but we actually do a dual transfection method because we got a helper plasmid from the Russell lab called PDGM6, and this is an AAV6 that has all those components in one plasmid, and you can obtain from the University of Washington with an MTA if you contact the Russell lab. If you don't want to file an MTA, there's a PDP6 sold by the plasmid factory that we've shown that is equivalent as the PDGM6. So once you have all your plasmids, you would transfect within HEC cells. We do about 10 plates, plating 15 million cells overnight, 15 centimeter plates. And then you allow 48 to 72 hours for replication and packaging within these cells. And then once that time comes, you harvest the cell lysate and you freeze thaw to release the virus. And then you treat with benzonase to release all the plasmids that were remaining within the cells. Following that, you're going to be doing harvesting and purification, which we recommend in the lab to get rid of empty capsids. And the reason we say that is because AAV production is not very efficient in the lab, and so you get about 10-fold more empty capsids than you do full capsids. And so we use the iodixinol purification method. And so to do that, it's just shown out here, and this is from a Nature protocol. You basically build gradients of different concentrations of iodixinol, so 15, 25, 40, and 56. And then you add your virus slowly on top. And then for ultracentrifugation for 130 minutes for 48,000 RPMs, your virus will go through these gradients and will leave protein at the top and cell debris at this layer between the 25 and 40. And then your full capsids will be between your 58 and 40. And over here on the right is showing a GFP one I made. And so this is a good sign. This means that the gradient ran and the GFP expression was so high that you likely have a lot of virus being made. And so that completes how to make the donor and how to make the AAV. And I just wanted to put this up here because these are the two electroporators we use. So this is the way to be able to deliver the RNP complex within the cells to make a double-strand break. So we use the Lonza 2B, which is the program U14, or we use the Lonza 4D, which is using the program TZ100. And so hopefully that would get us up to speed on generating gene targeting reagents. And so now I'll go through performing the actual gene targeting experiment. So this is kind of the outline that we use for the experimental design and analysis. And so the first part is cytokine stimulating your CD34s, either freshly purified or thaw frozen. The next would be to electroporate a pre-complex Cas9 RNPs and then transduce with the AAV6. And the following of your analysis will be analyze your HR efficiencies. And if you're interested in looking at stem cell function or progenitor cell function, it would be to initiate NSG transplants. And so the big thing we found, as long as others, is if you 2 days overnight culture at low density is key. So the reason I say that is because there's been a few papers from one in Toronto and one in Montreal and one in Toronto showing that if you use this fed batch system, you can get rapid expansion of human hematopoietic stem cells. And this, they hypothesized, controlled the inhibitory feedback signaling. And so the low density I describe here is less than 500,000 cells per mil and changing media every 2 days. And so the key is not to get over 1 million cells per mil or your viability starts to drop. And I've put all our cytokines within this text box over here. And maybe you could take a picture, but these are the cytokines and the catalog numbers in case people are interested. So the key is the culture at low densities. Another thing to do is to culture for 2 days before targeting. So the Naldini lab 3 years ago came out with a paper in Nature showing that if you culture the cells for 48 hours compared to 24 hours, in this case using with or without SR1, you could see more GFP positive cells within the CD133 positive, which is a stem cell marker fraction. And we have actually repeated this data. So culturing the cells for 2 days as opposed to 1 day helps for homologous recombination frequencies. So once you culture the cells for 2 days overnight at low density, then you are ready to target your favorite gene of interest. So I thought I'd just do this as a text box to go through the steps. And so this is actually the nitty-gritty. You know, this is how you do your experimental outline. And so what we usually do is count the cells to determine the number of experimental groups from replicates. A good marker is that the cell should grow 1.5 to 3 times in 48 hours. And really the more divisions, the higher the HR you get is an association we find. So the more times your cells divide before you target them, the better HR you will get. Then you have to determine if you want to do it in a strip format or a cuvette format. Within the strips, you do it in 20 microliters of solution, and we can do about 100,000 to 1 million cells. In the cuvette, you could do about 500,000 to 5 million cells. Then you would complex your modified guide with your SP-Cas9 at room temperature for 10 minutes, or you could do it at a thermocycler. And we use 150 to 300 micrograms per mil of Cas9, so that's per mil of nucleofection solution, as a 2.5 to 1 ratio of guide to protein, so that would come out to being 80 or 160 micrograms per mil of your modified guide. And I've shown an example here. You would spin your cells down for 300 G at 5 minutes, and then you aspirate and resuspend a nucleofection buffer. We use the P3, but we also have a recipe for a home brew that works well. If you want to contact me, I can send it to you. And then you combine your cells in your buffer with your RMP complex, and you pipette and your cuvette, and then it's ZAP. So ZAP is the electroporator using your program. And then you add fresh media, so the cells are now at 250,000 to 500,000 cells per mil. And one of the really critical things we found is that you need to add your AAV post-electroporation. And so up here is showing a graph when you add the AAV before 48 hours or 24 hours before electroporation, and then once you electroporate them, adding the AAV on after. And we get about a 4-fold increase. And we really think that is because electroporation is actually boosting AAV6 transduction. So showing here is your hours exposed to cell complementary, in this case AAV6, and no electroporation versus electroporation. This is the big difference. And another big thing is you want to make sure your AAV6 is less than 10% of your media volume. And so one of the points, the tips, is I think you really need to empirically determine the MOIs necessary to give you the highest HR with minimal toxicity. So in our hands, it's 50,000 to 200,000 multiplicity of infection using qPCR of ITRs. You really want to keep adding equal volume of media, so that's that kind of fed batch approach to keep the cells dilute. It promotes viability and it promotes their growth. And then next you would be checking targeting efficiencies, viability, and stem cell and progenitor function. So that gets me to this last part. Now that we targeted our favorite genes, how do we analyze what the efficiencies are? And so over here is just showing what a schematic about what this would look like once you target a gene for homologous recombination. And so knocking an SFFV reporter driving GFP into the beta globin locus, this is what it would look like afterwards following homologous recombination. And one of the things early that we noticed was that when you added a nuclease a nuclease and AAV, as opposed to just adding AAV alone, you saw a population that came up, which was presumably, we thought was a population that underwent homologous recombination, and in fact they do. And so a great way of analyzing GFP targeting rates you can do as early as day 3 or day 4 is to gate on this population, and that comes out to be in fact almost identical to what you get if you let the cells dilute out the episomal AAV. And so this is showing a correlation of your day 4 GFP high numbers versus your day 18 GFP percentages after you let the cells dilute out episomal AAV. And I wanted to put another thing on here too. You can do the same thing and figure out your HR rates in hematopoietic stem cells, at least immunophenotypic hematopoietic stem cells, by co-staining with hematopoietic markers and looking at the GFP high populations. It's kind of a quick, easy, cheat way of looking at your efficiencies in vitro. Another way to analyze your HR rates is on the DNA level by in and out PCR. And so quickly, we do this on methylcellulose clones. And A is showing a locus would be without integration. And so that we designed primers to get an 800 base pair PCR amplicon. And then a locus with integration, you'd have one primer that binds within your region that you targeted and one that's without the region that you targeted. And so this is what a representative gel would look like. This is what a wild type or indel, one that didn't have HR clone. This would be a biallelic because it didn't amplify the wild type locus, and then this would be a monoallelic clone. So it's also a good way to look at your monoallelic rates. Another way to do it is to by ddPCR. It's a similar strategy, so in and out PCR. And all you have to do is have one probe primer mix that's specific for your HR, your intended targeted locus, and one that's for your reference that's not your targeted. And you can multiplex this as long as one probe has hex and one probe has FAM, and this is showing a representative. You can also do it for SNP changes, and so that the concept is doing nested ddPCR for that, and it works great. I also recommend analyzing viability, and so for that I do PI and annexin. And so 1 or 2 days post-targeting, you do a co-stain of PI and annexin, you can see cells that underwent necrosis, underwent apoptosis, to die, and other ones that early apoptosis. And then while you're doing this, you can also co-stain for CD34 expression. You can do methylcellulose colony formation and look at HR frequencies, and then you can also look at HSC function. So this is the last thing I just wanted to show. I know my time is up. And so this is a representative image of what it would look like if you targeted true hematopoietic stem cells using this method. And so we use NSG mice, females, they can have higher chimerism. And then when the T cells, they develop week 16 to 24, B cells develop week 8 to 12, and myeloids develop early. So it's showing our gating scheme. We do HLA and human CD45, and then off of that we stain for B cells and we stain for myeloids to look for multilineage, and for CD3, looking at T cells, we look for CD3, and then within those gates you can look at GFP percentages. And with that, I'm just kind of put it up here as a text slide, kind of the take-home points. And so what we do is we screen synthetic guide RNAs and cell type of interest now. So we routinely screen these small scale 1 nanomolar from Synthego. So we go straight to the cell of interest as opposed to testing them in cell lines before. Then we also use highly purified high titer AAV6. And so we purify it either by idixanol or cesium chloride gradients, or you can also use some column purification methods. It's essential to transduce the AAV6 less than 1 hour post-electroporation. Ideally, we do it immediately, less than 15 minutes. And really, the cell culture conditions are critical. You know, this is biology of stem cells. And so culturing them at low densities, maintaining them often, never letting them get above 1E6 because their viability will drop. And when you transition from small scale to medium scale mouse studies, it's a good idea to test the surface area conversions. And in the beginning, you want to include mock electroporated AAV-only samples and RNP-only samples to establish your viabilities. And if you do have toxicities, then you can see where that's only coming from. And I highly recommend test hematopoietic stem cell function early and often once you start doing these projects. And you can expect a 2 to 4-fold drop in vivo from CD34 in vitro efficiencies. And just want to thank the Porteus lab and the collaborators and everybody that helped with this protocols. Of course, it wasn't me, it was a big team effort. And I'll take questions at the end if anybody has any. Thanks a lot.
Thank you so much for your presentation, Dr. Dever. Very informative and definitely nitty-gritty details. We appreciate it. Now I have the privilege of introducing our last speaker, Dr. Michael Gundry. Dr. Gundry received his Bachelor of Science degree from the University of California, San Diego in 2008. He then worked as a research assistant in Dr. Vige's lab at Albert Einstein College of Medicine, where he developed the first assay for whole genome amplification and genotyping of single mammalian cells. In 2011, he joined Baylor College of Medicine MD PhD's program and is currently in the laboratory of Dr. Margaret Goodell. His thesis work is focused on the role of DNMT3A and MPN1 in normal and malignant hematopoiesis. In addition to the mouse models, Michael has previously worked extensively with primary human hematopoietic stem and progenitor cells and has developed assays for efficient CRISPR editing in these cells. After completion of his program, Michael plans on pursuing a residency in pediatrics and a fellowship in hematology oncology with the long-term goal of working as a physician scientist. I will now turn the presentation over to Michael.
Thank you, David, for the kind introduction, and I want to start by thanking the ISEH organizers for giving me the opportunity to present. So like David just mentioned, my name is Michael Gundry, and I'm a sixth-year MD PhD student in the Goodell lab. So our lab classically has been a hematopoietic stem cell biology lab. We've done a lot of work in mouse models, and in the last 5 years have been focusing on a gene called DNMT3A. When I joined the lab, I was looking to see if we could expand our work into working with primary human cells. And with the advent of CRISPR and its increasing ease of use, the timing was kind of perfect for this. And so that's kind of what I'm going to be talking about today. Just the title is Fast and Efficient CRISPR-Cas9 Mediated Gene Editing in Human Hematopoietic Cells. So I'm going to start by kind of going through brief history. A lot of the early protocols available for CRISPR involved either plasmid-based or lentiviral-based delivery. And the early systems were bipartite, where you needed a separate vector for Cas9 and for the sgRNA or the guide RNA. But those were soon replaced by single vectors where both components were encoded, as seen here, on a single plasmid or a single lentiviral vector. So I was saying that a lot of the early protocols were plasmid or lentiviral-based delivery, and some of the drawbacks of these protocols were that cloning was required, which is kind of an everyday lab assay, but can take some time. And if you're working with a lot of guides, trying to screen 20, 30 guides at the same time, this can be somewhat cumbersome. You did need to deliver two elements, the Cas9 and sgRNA, and often because of the size of the coding sequence of Cas9, this led to very large constructs which were more difficult to transfect and transduce. When actually trying to transfect and transduce primary hematopoietic cells, a lot of the issues that Dr. Dever was mentioning related to toxicity became issues. And so not only did you have high toxicity rates when trying to deliver to primary hematopoietic stem and progenitor cells, but you also had low efficiency even following the use of selectable markers. So most of the early work was unable to get past a kind of threshold of 20 to 25% gene editing with these plasmid or lentiviral-based delivery approaches. And so a number of groups, a lot of groups in gene therapy, as well as our lab, started exploring alternatives, and one that we stumbled upon and began using in our lab and now use for all our projects in our lab was ribonuclear protein-based delivery. Now this is delivering the raw components of CRISPR, the actual Cas9 protein and in vitro transcribed sgRNA. And in addition to the ease of use, where, again, Dr. Dever was mentioning that they actually now buy most of their sgRNAs from a number of different vendors and have modified sgRNAs. Again, they buy their Cas9 protein, so the actual hands-on work related to construction of plasmid or lentivirus is somewhat mitigated. In addition to that, the efficiencies of these RNP-based delivery approaches in our hands and in many others' hands were much, much higher. And so just to kind of describe our workflow, and I'll go into a little more detail in a second. On day one, we actually, when we're screening guides, we make them ourselves using in vitro transcription, and I'll again go through some of the details of that in a second. And with in vitro transcription, we order primers that are specific for each of our guides. They have a T7 promoter, which allows for their in vitro transcription following the generation of an amplicon. So on day one, we do sgRNA synthesis. On day two, we form our RNPs by incubating our Cas9 protein, which we are buying from vendors similar to those that Dr. Dever mentioned. We incubate our Cas9 protein with our in vitro transcribed guide, add our cells, whether these are hematopoietic stem and progenitor cells from a human or mouse, or cell lines. We add those and electroporate, and on day three, we're able to assess the knockout efficiency and begin our in vivo or in vitro experiments. So I just want to give a brief overview of our RNP protocol. You can design your guides from a number of different online algorithms. Each algorithm has a slightly different scoring approach for both on-target and off-target efficiency. In our lab, we use a algorithm called CRISPR-scan. It's nice because it has a nice user UCSC browser functionality, which I think is really useful, especially if you are trying to target specific exons and trying to be able to visualize these exons while you're choosing what are the appropriate guides. So after designing guides and ordering our primers contain, like I said before, a T7 promoter sequence, which allows for the in vitro transcription, and then our target sequence, and this is the specific sequence in the genome that you are targeting and will be cleaved by the Cas9 endonuclease. And then they contain at the 3' end a 15 base pair universal scaffold reverse. This is the first part of the sgRNA scaffold, which is conserved across any sgRNA that you're designing. We have a universal scaffold reverse primer, and we just do overlap PCR. So we use this primer for all our guide synthesis, and this is the custom primer we order for an individual guide. Now, normally, and I'll kind of talk about some of the workflow when you're targeting a gene later, but normally when we have a new gene we want to target, in this case, let's say PTPRC, which encodes for CD45, a cell surface receptor, we will design a number of guides per exon, and we might target 3 or 4 exons as a first trial. You can easily make 10, 20 guides in a short amount of time, and actually scaling it up to 20, 30 versus doing 1 to 5 is not that difficult. So once you've finished your overlap PCR, you now have a double-stranded DNA template, and this is the template that's used for in vitro transcription. And the in vitro transcription, again, you can do almost directly after the PCR, and it takes about 4 hours. You can run it overnight if you want, if you're trying to save some time. Now, following in vitro transcription, you purify your RNA, and then we form our RNPs by incubating our in vitro transcribed RNA with Cas9 protein, and we use a very similar ratio. We use a 3-to-1 molar ratio of sgRNA to protein, and this, for those who are working with micrograms, for us, when we're doing neon electroporations, 10 microliter electroporations, we're using 1.5 micrograms of the Cas9 protein and about a microgram of sgRNA. And you add your RNPs to whatever cell type of interest you're working with, and electroporate. And Dr. Dever was talking about using the Lund instrument. We use the neon instrument, but have also used the Lund instrument. I think groups have found very high efficiencies with these, with both of these protocols. And in my hands, actually, the Lunds are a little bit more consistent because I think the user error, especially at the very end when you're actually performing the electroporation step, the user error is a little bit lower. But I think both will work for your experiments. And finally, we assess knockout efficiency using a surveyor assay, Sanger sequencing, amplicon sequencing, or flow cytometry. We use amplicon sequencing a lot, and that's because we do a lot of RNA and chip-seq, and so we're constantly running high-throughput sequencing runs. And how this works is we're just performing a PCR on our targeted region. and spiking in however many samples we have. Sometimes we'll screen 10 guides, and so if we're working with a single cell line, that means we'll screen 10 guides, we'll have 10 amplicons, and we'll just throw those into a high-throughput sequencing run in order to actually get a really accurate estimate of what our indel spectrum is and what our overall disruption frequency is. And this is just showing an example output here. So this is the wild-type sequence for this locus. It was unedited, and 12 to 13% of our alleles had this wild-type sequencing. This wild-type sequence, indicating that the other 87% or so were likely targeted. And this is just showing some of the most frequent indels. We saw a 2 base pair deletion, a 2 base pair deletion, 1 base pair insertion, etc. So that was kind of a brief overview. Now I kind of want to go through some results. And those are going to be broken down into 2 major targets, actually. The first part was made up the majority of our paper published in our paper published last year in Cell Reports, and focused on targeting a cell surface receptor. And the second half is going to be on targeting DNMT3A, which is the gene that our lab is actually interested in. So here I'm showing 3 AML cell lines, and this was some of the early optimization work we were doing. So I'm showing 3 AML cell lines, and we started by targeting a cell surface receptor that's present on all hematopoietic cells, which is encoded by the gene PTPRC and is CD45. And here I'm showing really efficient knockout, and I'm showing flow cytometry data showing the normal expression pattern across a population of cells in Cas9 only, or untreated cells, and you can see that here in the light blue line. And then the expression of, again, this cell surface receptor CD45, 3 days after targeting with our CD45 guide. And you can see here the knockout efficiency is almost 100%. In this case, there's not even really an observable population of wild-type cells. In some of the other cell types, you can see some residual wild-type or maybe heterozygous cells where you're actually targeting one of the 2 alleles. So this is in 3 AML cell lines. We also looked at trying to target multiple cell surface receptors at the same time. Here we targeted both CD19 and CD22. Again, here you see a population of cells where you only targeted and knocked out CD22. Here a population of cells where you only targeted and knocked out CD19. You can see here's the normal expression pattern of CD19, and here's what we see now. And this is a large population of cells. It was actually 70% or so where you targeted both CD19 and CD22. And this actually indicates that you were targeting both alleles of CD19 and both alleles of CD22. We then moved to primary samples and looked at activated T cells and saw similar knockout efficiencies. Actually, with the T cells, we saw some new populations by flow cytometry. Actually, some of these were in-frame deletions where you had a partial loss of function and therefore had some residual binding to our antibody in flow cytometry. And then here is showing primary NK cells, in which, again, we also saw really highly efficient knockout compared to this Cas9 only treated sample. But we were particularly interested in working with human hematopoietic stem and progenitor cells. And so a couple of different sources of these were mentioned by Dr. Durr in his last talk. We focused on umbilical cord blood samples, which we had access to through a MD Anderson cord blood bank. And the brief workflow, we would obtain these cord bloods, we would enrich for CD34 positive cells, which contain the most primitive hematopoietic stem and progenitor populations. And then we were culturing them for between 36 and 48 hours. I'll give some tips later on that. And following a culture period with cytokines, we performed a stem electroporation. Again, this was targeting CD45. But I also have CD34 here just to show that these cells are not losing potency following electroporation. So you can see the normal population of, this is a sorted population of CD34 cells. The normal expression pattern of CD45 in Cas9 only treated samples here. And you can see a large percentage of cells. In this case, it was about 80% of the cells which had lost CD45 expression 24 hours to 48 hours after electroporation. We tried a number of different targets. Again, our lab's interested in DNMT3A. got a number of different targets, and I'm going to focus a little bit on this exon 10 later, but we saw really high rates of indel frequencies or disruption frequencies across all our targets, between 70 and 90%. So one thing we were really curious about and thought was an important validation was whether these cells, upon electroporation and CRISPR treatment, whether these cells would lose their multi-lineage engraftment capacity, lose their stemness. And so this is showing samples, and in this case it's cord blood samples where we enriched for CD34 cells, we knocked out CD45, and then we engrafted these into immunocompromised NSG mice. And this is showing 4 months post-engraftment. And you can see here a population of human cells, both in the bone marrow and the spleen. This population here, which is mouse cells, does not contain either the human HLA-ABC marker or human CD45. And so you can see about 50% of the cells in this mouse, this is a single mouse, were engrafted human cells. And this is a cord blood which we split into Cas9 only and CD45 sgRNA treated. So following sgRNA addition, what you saw is a third population. And this is a population that is a human cell which has human HLA-ABC, but is not CD45, as that's what we were targeting. And you can see here the efficiency in the human cells is about 50% in both spleen and bone marrow. Again, this represents that CD45 negative population. Importantly, when we looked at the CD45 negative population and the positive population, we didn't really see any difference in the cellular phenotypes in the percentage of myeloid and lymphoid cells, indicating that in this case, neither CD45 loss or the actual electroporation and CRISPR treatment was affecting the multi-lineage engraftment capacity. I'm going to switch over to the MT3A, and this is a little different because instead of targeting CD45, and we tried multiple guides and we were getting really efficient knockout with each of our guides, with DNMT3A we really took a brute force approach and designed 30 guides spanning all the conserved exons. And I say conserved, there's 2 major isoforms of DNMT3A, and so here we were targeting exons that were conserved across both the short and the long isoform. We actually electroporated multiple guides together, so we were trying to create a combination of deletions, and some of these deletions would be framed, some would be out of frame, kind of similar to what, again, Dr. Bauer was talking about in his last talk. You don't always have precise deletion between your 2 cut sites. So here, because we were in some cases using 4 guides per electroporation, you could have a number of different outcomes, and this is just showing the different types of deletions you can see. If you did a PCR across the locus, you would expect to see multiple different bands representing these different deleted alleles in a population. And that's what we saw. Here, this is just showing PCRs of 4 guides for exon 7, 8, 9, 10, 11, 12, and 13, 14. And you can see multiple bands representing different alleles, and those alleles were derived from these different combinations of deletions. When we looked at the protein level for these cells, we saw that all guide combinations were significantly reducing DNMT3 protein. This is showing Cas9 only, and this is showing, again, these 4 different population cells. So this is the bulk population, and we're seeing almost complete loss of DNMT3A. And when we did amplicon sequencing, we found that especially with this DNMT3 exon 10 targeting, we were seeing disruption frequencies approaching 90%. So this is what we proceeded with for our in vivo studies. And similar to what I was showing with CD45, when we looked at the engraftment capacity, or the engraftment of Cas9 only versus the DNMT3A knockout cord bloods, again, each of these cord bloods we split in 2 so that we have an internal control. We didn't really see any significant difference between the 2, and that was good. Same thing when it came to the different lineages. We actually had really low, for some reason, really low myeloid engraftment in this set of experiments, but we didn't see any significant difference between the Cas9 only and 3A knockout treated samples with respect to percentage of B cells, T cells, and myeloid cells. And this is showing a amplicon of that focus. You can see here is our wild type. This represents the 300 base pair, the 300 base pair PCR product we would expect. And here we actually use, and I should have mentioned that earlier, we use two guides, a combination of two guides targeting exon 10, and therefore we expected a population of alleles to have that exon 10 deletion, which we saw. When we did amplicon sequencing, again, this is following engraftment and recovery of CD34 positive cells. We saw allele knockout or disruption rates between 70 and 90%. Now we took these 1, 2, 3 cord blood knockouts and their matched control samples and started to look at some of our molecular phenotypes. This is really what I think holds the most promise in some ways for, at least from a basic biology standpoint, for these new and novel CRISPR approaches in hematopoietic research, is that in a way it's not that it's replacing or going to replace mouse genetics, but it's a supplement or an alternative to some mouse genetics approaches in which you can actually work with human hematopoietic cord stem cells and begin to explore molecular and cellular phenotypes following engraftment. And these are kind of some in vivo phenotypes. So this is kind of a complicated slide, but here we have our 3 on the left. We have our 3 cord bloods, which were treated with just Cas9, and our 3 cord bloods, which had our exon 10 knockout. And this knockout efficiency was, again, between 70 and 90%. And what we're looking at here is whole genome bisulfite sequencing data. And this type of sequencing data allows for the single CpG resolution. So each of these blue bars here represents the methylation status at a single CpG site. And you're able to look across the entire genome and see what regions are fully methylated. This blue bar here would represent 100% methylation, and what regions are 0% methylation, which would be like a region here. Now, with 3A loss in the mouse, we saw focal hypomethylation in some particular regions, and that's what we were looking for now working with these human engrafted progenitor cells. And that's what we saw. We saw with a couple key genes involved in acute myeloid leukemia and a number of other hematopoietic malignancies, such as the HOX-A, HOX-B, and MEIS1 cluster, we saw significant hypomethylation at some focal regions. Here it's not as significant at the HOX-A cluster, but you can see much more significant loss of methylation at the HOX-B cluster, as well as in MEIS1. And again, this is with a bulk population of cells, so it was really dependent on a really high efficiency knockout, which we were seeing between 70 and 90%, in order to be able to run this kind of a molecular phenotypic assay and actually see results from the bulk population of cells. So that's kind of the data, and I just wanted to finish up with some tips for efficient gene disruption. So the RNP approach works really well, and I showed a little bit of data from both cell lines and primary samples. We've actually done some work with primary leukemias as well, and it looks really promising. So it appears as though for all suspension cells, including both cell lines and primary samples, this approach is one that you should definitely look at. When one's planning experiments to test for the effect of loss of function of a gene in a cell line or primary sample, I think it's really important to start out by targeting the appropriate exons. And the two important factors here come down to conservation across all isoforms. You have to make sure that you're not targeting an exon, even if it's an early exon, you're not targeting an exon that's specific to an isoform that's not even expressed in your cell line. And the second is to target early exons. Really, you want to avoid the terminal and penultimate exons, which will have somewhat unknown consequences. They often don't result in nonsense-mediated decay. Instead, a truncated protein product can be produced with an unknown function. We've had a lot of success screening multiple sgRNAs in combination. And again, we're designing, if we have a new gene target, which we've now looked at between 50 and 100 genes, and this strategy has been really effective. We're designing 10 to 30 guides. We're screening those guides using in vitro transcription, and then adding them in combination, targeting 2 to 3 guides per exon, looking at multiple exons and seeing which ones result in both high disruption rates and then loss of protein product. It's really important that you're doing Western blots, or at least if there is a good antibody available, you're doing Western blots to make sure that you're actually leading to loss of the protein product. We often do, and I know that some groups may not and may disagree with this, but we often do use a suspension cell line like K562 or HL60 to screen for efficient sgRNAs before working with our core blood samples. We've actually found high concordance between the efficiencies in our cell lines and primary samples, and therefore that's why we've decided to proceed with this. Like I said before, assess protein levels when possible, and we often use, and I don't actually show too much data from that here today, but we use a lot of control sgRNAs. So again, because we're screening with 20 to 30 guides that we're in vitro transcribing, we often have some guides which have toxicity that might be due to off targets. You might be targeting 20 or 30 regions in the genome, or it might be due to poor quality purification of that IVT product. And we're not really sure. I think there's both unknown and known factors related to why some guides in our hands are more toxic than others. So in those kind of settings, especially if you're doing in vitro or in vivo assays with hematopoietic progenitor cells, you have to make sure that you're using a lot of control, a number of control sgRNAs to make sure that the phenotype you're seeing is not one related to just guide toxicity. As Dr. Dever was talking about before, it's important to keep culture time short if you're working with primary samples, either both T cells, but more importantly with your hematopoietic stem and progenitor cells. Definitely we've had a couple instances where we use guides like CD40, our guide for CD45, for a number of projects. We're using it as a control for all our experiments. And in those cases, it's really useful to actually buy from both Synthago and TriLink, the same two companies you mentioned, because they do have a lower toxicity compared to your IVT-produced guides. And in general, it's you can actually ramp up your scale now with Synthago to much higher yields. You can use it for 1,000 or so experiments. So just want to finish by thanking the Goodell Lab. I did a lot of this work with Lorenzo Brunetti, who's a MD in Italy, as well as our collaborators at Baylor, at the Feigen Center, and at MD Anderson. Thank you.
Michael, thank you so much for a wonderful talk. It's really proving to be both interesting and exciting, but also a challenging field to be sure to be a part of. We can now open the floor for questions and for a bit of Q&A. I would again like to remind everyone in the audience who would like to submit a question to our speakers, please to type it into the question or chat boxes on the screens. And I guess I can start us off quickly with a question to Dr. Bauer, actually. You mentioned that you get a mixture of products for every single guide, and I was wondering, in terms of the perspective of design and experiment and designing an extended sort of experiment like a pooled screen, how do you take those mixture of editing products into account? What is some considerations that a person could be thinking about?
Right. So one of the key features of a pooled screen is maintaining representation of the library throughout the experiment, and so that would be for any one guide RNA in the library, how many individual cells are expressing that guide RNA. So generally we aim for about 1,000-fold representation. So this is really a key consideration, because if it's a large library, let's say if it's one of the genome-wide libraries with 6 guides per gene, it might be over 100,000 guides in the library, and if you want to represent each guide in 1,000 cells, it might be over 100 million cells that at minimum need to be maintained throughout the experiment. And so in part, that the representation addresses a number of issues. One of them is the heterogeneity of editing outcomes. So if a given guide RNA, if it's targeting, well, first of all, if the editing efficiency isn't 100%, there may be some unedited alleles, and even if it edits, there's a variety of outcomes. If it's coding sequence, there could be frame shift versus in-frame alleles. The in-frame alleles may preserve function. If it's targeting non-coding sequence, depending on the nature of the indel, it may or may not have functional consequence. So by including more guides, more cells per guide, one can account for that. The other thing is it depends on what your assay is, and so if it's kind of an assay where something gets turned on, cells survive, a reporter gene is activated, it's very sensitive to find rare events. If it's more of like an off assay where cells drop out, that it may be harder to detect rare events in the population. So I think part of it is designing an assay that's suitable for the type of outcomes you're anticipating.
Of course, thank you. And now a question for Dr. Dever. In terms of the HSC transplantation data that you published in your paper, I was just wondering if I could ask you a general question in terms of the AAV and the general sort of characteristics of toxicity. How would you evaluate the different sources for HSC sort of related toxicity, and in the context of transplantation, how do you address them, given the fact that you recommended a culture period of some length?
You mean toxicity for the different CD34 sources?
Yes. No, I mean the toxicity for CD34s after treatment.
Oh yeah, so we have real good viability after all of our reagents. Again, we use the same strategy as Michael was pointing out with the RMP. And once you add the AAV, I would say probably 80% of the cells are PI negative and Nexin negative. So live cells, viable cells, and it always depends on the quality of your reagents and how much virus you're using. For the culture length time, we try to keep it under 5 days for our transplants. So if we're going to sort targeted cells, we can see the MFI shift max around day 3. And since we do it 2 days before, we keep the culturing around day 5. We have tinkered around with doing the Toronto approach with using UM171 and culturing the cells for 2 weeks. And it actually does work. The only thing there is that they lose potential, so you have to use a vast amount more cells. So for example, if you put in 1,034,000 at day 5 and you got 20% engraftment or 30% engraftment, you probably need to use, 5 times that at day 14 in culture. So in our hands, day 5 seems to work pretty well for getting high engraftment. And for us, it's always a numbers game. Like, these are experimental mouse models, and you can always max it out to get the most engraftment you need for your endpoints. Of course, that's a different concern when it comes to translational approaches. But for us, the toxicity seems to be relatively low.
Wonderful. Thank you. Another question for Dr. Gundry. Our audience member asks, have you tested the efficiency of the RMP approach for homologous recombination gene targeting in your cells?
Yeah, so that's what Dr. Dever was actually presenting on. He was using the RMP approach. So we actually haven't worked with AAV6. We worked with single-stranded ODNs, which is an alternative. And it worked well in our hands. We were getting between 25 and 30% with some loci. I think there's definitely some variability with respect to your target locus, how far your guide is away from your guide site's away from your region that you're actually trying to edit. We've also worked a good amount actually with double-stranded HDR templates, and haven't actually used those too much in CD34 cells. We use them a lot in cell lines. And these are large double-stranded. Instead of a plasmid donor, we use double-stranded kind of donors we're making through PCR and through actually some combinations of Gibson assembly and then PCR. And we're able to get 1 to 2% efficiency in cell lines when it comes to GFP tagging of different proteins, et cetera. So I think there are multiple different approaches for HR, and you really can pick and choose depending on what your target and cell type is. I think for the CD34 cells, I think the AAV6 has kind of been shown to be the way to go.
Wonderful. Now, since we're running a bit low on time, I would like to ask each one of our speakers if they have any general thoughts or advices for our audience in terms of what are the main considerations in design of an experiment that they should address, a key control that you can think of. Starting with Dr. Bauer, perhaps.
Well, controls are always important. I think we are seeing that depending on the context, that DNA cleavage itself can be have some biologic significance to cells. So a target at a neutral locus can actually be probably the most equivalent control for any locus-specific effects that take into account that Cas9 has cut somewhere at the similar copy number as your target locus, and that the DNA repair response has been activated.
I guess
Dr. Bauer, we seem to have a small problem receiving you.
I heard him.
I heard it too. Yeah, too.
Well,
Okay,
maybe
I can follow up. Yeah, I think that Dr. Bauer was right on with the controls. That's what we've moved towards for all of our experiments. We use, again, it's ideal to use a neutral locus, either a non-coding region or a gene that's not expressed in your cell type. But you have to make sure that your actual cleavage efficiency with that site is high. I think that for us, the direction that we've moved with all our RNP approaches have been to screen guides with IVT, and then those in which we know we're going to use for follow-up experiments, order those guides from Synthigo. Our experience with Synthigo, I don't know if Dr. Dever has worked with any of their non-modified guides, but our experience is that the non-modified guides are pretty poor and actually have much lower efficiencies than our IVT guides. Again, our IVT guides have no modifications. So if you're going to go with the Synthigo or the TriLink route, I would definitely use the modified guides, which have really high efficiencies, similar to what we get with IVT.
Yeah, it's something in the air out here, so we always use the modified guides. We can't go back.
Yeah.
But I just had, I guess, two things. One thing is, you guys, the audience out there is taking our word for it. But if you have any questions, I would say just do it. So if you want to compare AAV6 versus SSODNs, I would compare them side by side and with your own hands, who you trust, and see if you get the same results. If you want to try an approach to increase hemoglobin, like Dan Bauer saying, try that approach versus other approaches, like head-to-head, in my experience, is always the best way to go. Then you just follow the data. And then secondly, choosing your CD34 source, in my case, for translational approaches, was always huge, because you can kind of be led astray if you always use cord bloods, but then when you're going to translate, you're going to be using mobilized peripheral blood in an adult patient. And so always just keep that in mind about what your end goal is, whether you're a graduate student, a postdoc, or you have your own lab.
Thank you so much. I would like to mention that the rest of the questions, we're running a little low on time, and so the rest of the questions will be funneled to the presenters for responses via email. Excellent. So I would like to thank everyone for participating in today's webinar. And I would like to extend a special thank you to our presenters for today's session, Drs. Bauer, Dr. Deaver, and Dr. Gundry. For the audience and attendees, if you could please complete the evaluation for today's event that you will receive via email. Your feedback and input, of course, are important and appreciated as we further develop ISEH webinar programming. I would like to thank you all again for attending and wish you a wonderful, wonderful day.